Hello dear students. We have already done this topic carbohydrates in the live class, but because of poor network some of you could not get it clearly. Therefore let's study it once again. So these carbohydrates, as I told you, they are the biomolecules which are very important. Before going into the details of these carbohydrates, let us first quickly study about their properties. They are commonly called as sugars. The sugar word comes from saccharides. Saccharum is the Greek word for sugar. So these are sugars. They are sweet in taste. Some carbohydrates may be very sweet in taste like fructose, glucose. Some may be less sweeter in taste like starch. Okay. So but they are sugary. They are sweet in taste. Second, the chemical composition. They are made up of carbon, hydrogen and oxygen with a general formula CnH2O n times, where n ranges between 3 to 7. Also they are energy sources. All form of energy are there, like in the form of glucose, starch, cellulose. All these are energy sources. And they are optical isomers. One more thing, they are chemically polyhydroxy aldehydes or ketones. So this means they have many OH groups. Okay. Now before going into the details, let us first classify them. How do we classify the carbohydrates? Therefore, these carbohydrates can be classified into three categories. These three categories are monosaccharides, oligosaccharides, and polysaccharides. Okay. These monosaccharides, they may be triose, tetrose, pentose, hexose, and heptose. Because we have studied that n ranges between 3 to 7. So the smallest carbohydrate are triose. The example, very common example is glyceraldehyde. They are triose. Tetrose example is erythrose. Pentose, ribose and deoxyribose are the examples for pentose. Likewise, glucose, fructose and galactose, they are the examples for hexose. And heptose are pseudoheptose. Now, as I told you, the general formula is this. So, you can write the general formula for trioses. Trioses will be C3H6O3. That's why we say C3 sugars. Okay. Same way, they will be C4H8O4. They will be C5H10O5. This is deoxy. One oxygen is less. Therefore, it will be C5H10O4. Now, hexoses will be C6H12O6. And heptoses will be C7H14O7. Okay. So, these are the monosaccharides. Monosaccharides are the micromolecules. They are very small units. They are soluble in water, sweet in taste. Okay. And you will come across these terms when you will study about C3 and C4 cycle in case of photosynthesis. Also, when we will study about TCA cycle, Krebs cycle in respiration, then also you will come across these terms because these will be the intermediates in those cycles. So you will have to apply these terms there. Okay. So these are different levels of monosaccharides. Fine. So the first type of carbohydrates are monosaccharides. I will come to monosaccharides again into detail their structures. Then second category are the oligosaccharides, where there may be 2 to 7 monosaccharide units attached. Okay. So the most common ones are the disaccharides. In disaccharides we study maltose, then sucrose and lactose. They are very common ones. Okay. So you know maltose are made up of 2 units of glucose. Glucose plus glucose. Likewise sucrose. They are made up of 1 glucose plus 1 fructose. Then lactose, they are made up of 1 glucose plus 1 galactose. Okay. So these are disaccharides. How are they combined together? There is a specific type of bond which is called as glycosidic bond. We will study about the glycosidic bond. Then comes the polysaccharides. Polysaccharides, they are many. When many n number of monosaccharides are attached together with the help of glycosidic bonds, then the polysaccharides are produced. And these polysaccharides again can be of 2 types, homopolysaccharides and heteropolysaccharides. Example of homopolysaccharides are starch, glycogen and cellulose. Heteropolysaccharide examples are N-acetyl, glucosamine. This N-acetyl glucosamine, it is present on the cell wall of prokaryotes, bacteria. Okay. Here along with the glucose there is acetyl group and amine group is attached. Okay. We will come across to this structure later on. Let us first study the structure, molecular structure of monosaccharides. In monosaccharides, we will mainly study the structure of pentose and hexose. So let us see. In case of pentoses, one is ribose, other is deoxyribose. As I told you, the formula is C5H10O5. So the structure is like this. Like this. Okay. This is carbon position 1, 2, 3, 4, 5. Now the structure of deoxyribose, the formula is C5H10O4. There is a simple difference of only 1 oxygen. All other structure is same. Only here, instead of OH, there is only H. Otherwise, the structure is almost the same. H, OH, OH, H. Then CH2, OH, H. This way. Okay. So this is a these two are the structures of pentose sugars. They are the backbone for RNA and DNA respectively. Now let us come to hexoses. In hexoses, mainly we are talking about the glucose. Let us see the structure of glucose. Glucose, the formula is C6H12O6. This glucose, fructose and galactose, they have unique property. They may be present in aliphatic or in the aromatic form. So, the aliphatic structure is like this: CH2OH, CHOH, 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 CHOH and CH2OH, like this. This is the aliphatic structure. But whenever it forms the bond, it is present in the cyclic form. I'll draw the structure now. In this way. H, OH. This is the structure of glucose. Here it is carbon position 1. This is called 1 prime, 2, 3, 4 and this is 5 and this is 6. Okay. So these are the positions for the carbon. Now, likewise, the structure of fructose is almost same, but the difference is like this. It has the aldehyde group, whereas the fructose has the ketone group. This is the structure of the fructose. Here the carbon position 1, 2, 3, 4, 5 and 6. Okay. So these are the monosaccharides. Now we will study how the two monosaccharides combine together to form the disaccharide. And the bond which is there is called glycosidic bond. And the glycosidic bond, I will tell you, two types of glycosidic bonds: alpha glycosidic bonds and beta glycosidic bonds. Let us study the formation of the two bonds, that is glycosidic bonds, alpha and beta. So the bonds between any two carbohydrates are glycosidic bonds. So I will tell you about the alpha glycosidic bond and beta glycosidic bond. Let's just see how are they different. The difference is only in terms of the plane they are present. Now, suppose we are making two glucose molecules. This is one glucose molecule. Likewise, this is another glucose molecule. Okay, you know the positions. Likewise, here also, let's draw two glucose molecules. So, a bond is formed by polymerization or condensation. You know, when there is condensation, one water molecule will be removed. Likewise, when the water molecule removed is at the same plane, alpha type of bond is produced. When the water molecule lost is at the different planes, then beta type of bond is formed. Now let us see. You know the position. This is 1, 2, 3, 4. Okay. 5 and here there it will be 6 here. Likewise, this is 1, 2, 3, 4, 5 and 6 will be here. because here it will be the carbon here. Now the bond formed will be in this way. So this is 1,4 alpha glycosidic bond. Alpha 1,4 glycosidic bond. Here the bond form will be in different plane. Therefore, the bond formed will be like this. Here also, it is the same position. 1, 2, 3, 4. 1, 2, 3, 4. Okay. So, the bond is formed, but it is not alpha. It is beta 1,4 glycosidic bond. Okay. So, in this way, the bond formation is there. These are the disaccharides. Likewise, when many units they attach with each other, then polysaccharides are produced. Okay, so let us study the different types of polysaccharides now. We will study about the homopolysaccharides. Different homopolysaccharides. Different homopolysaccharides, I will tell you three main homopolysaccharides. One are the starch, glycogen, and cellulose. You will be amazed to know that all these three, they are made up of only glucose unit. All three of them are n number of glucose units. This is also n glucose unit. This is also n number of glucose units. Then how are they different? They are different only in terms of the bonds and branching. Let's see. The starch. Starch is actually made up of two types of homopolymer chains. One is called amylose. Other is called as amylopectin. These two chains are also just like the n number of glucose units. But the only thing is that this is unbranched, this is branched. Here, amylose is a soluble type of polysaccharide chain, whereas amylopectin is insoluble in water. Amylose is soluble in water and amylopectin is insoluble in water. So amylose chain, the structure is like this. Whereas amylopectin, the structure is like this. These are the branched structure. I'll just make this part. Okay. This part is nothing but just like there are glycosidic bonds, alpha glycosidic bonds like this. In case of amylose is like this and it's just a spiral. But whenever there is a branching, at the point of branching, instead of alpha 1,4 glycosidic bond, there is alpha 1,6 glycosidic bond only at the point of branching. Let's see how. We know that suppose here the branching starts. Here will be 6th position of carbon. I told you there will be 6th position of carbon. This 6th position of carbon will be forming the bond with the first position of another. And here will be the bond formation. And again, there will be the glycosidic bonds like this. So this portion, this will be alpha 1,6 glycosidic bond. Okay. So this is about the starch. Now let's come to the glycogen. Glycogen is just like starch. In fact, glycogen is called as animal starch. Glycogen has only one chain, but it is profusely branched. Like this is branched so much. This is made up of n number of glucose units and exactly like this. This is less branched, whereas this one is highly branched. Okay. Now let's come to cellulose. Cellulose is also made up of glucose. The only difference is that it is made up of beta 1,4 glycosidic bond, and there is no branching. Okay. Like the glucose and glycogen, they both can be digested by humans because we have the amylase. The amylase can digest the amylose chain as well as amylopectin chain. That enzyme, that is salivary amylase, we have got pancreatic amylase, intestinal amylase. So we can digest both the starch as well as glycogen. Okay. And the starch, when it reacts with iodine, it converts it to blue-black color because it holds the iodine in its coil. Whereas this glycogen is not able to convert it to blue-black color because it does not have any coiling structure which can hold it. Likewise, cellulose also cannot convert the iodine into blue-black color. Okay. So now cellulose. Cellulose is just like the starch only because it is also made up of n number of glucose units. The only difference you can see is the formation of the beta glycosidic bonds. So the bond formed are like this. In this way. Okay. So this was about the cellulose. So these were the different types of carbohydrates. Likewise, the heteropolysaccharide N-acetyl glucosamine, that is present in the cell wall of the prokaryotic cell. And this cellulose, it provides energy only to herbivores because they have basically the enzyme, cellulase enzyme, which is present in their rumen. They have to ruminate it. Okay. So this enzyme cellulase is present due to the secretions by certain microbes, and it can digest the cellulose to provide the energy. Whereas humans cannot digest it. We can only digest either starch or glycogen. The basic function of cellulose in case of plants is structural firmity because all the cell walls, they are made up of cellulose. Primary cell walls are made up of cellulose. Okay. So with this we have completed the different types of carbohydrates. Go through it in your textbook. In your textbook they are not given very systematically. In fact, they are given very heterogeneously. I have all grouped them together so that there is connectivity. So these carbohydrates, they form a very important aspects, not only for energy currency, not only for the energy source, but also as in the form of pentoses, they are the building blocks for the nucleic acids also, because when we will study about the nucleic acids in the next segment, we will study that the backbone of the nucleic acid, that is DNA, is having the monosaccharides in the form of pentoses, that is riboses and deoxyriboses. Therefore, they are very important. You will be studying their structure in your chemistry also in class 12. So please go through it, understand it, and even if you have after that you have any problem, you share it with me. Okay, thank you.